Hello, everyone. Thank you very much for joining us for the Huntington Society of Canada's April Research Spotlight webinar. My name is Alexander Maxon. I'm the Director of Research at the Huntington Society of Canada. Thank you all so much for being in attendance today as we learn more from Dr. Adam Hendricks, who's going to be presenting. So Dr. Hendricks works at McGill University in Montreal as Department Chair and Associate Professor in Bioengineering. Today he's going to present about his team's research on two aspects of Huntington, specifically the protein, I believe. So one is the role of Huntington and Huntington-associated proteins in coordinating intracellular transport, and then how interactions with RNA in the unstructured domains of Huntington modulates its phase separation. At HSC, we're very grateful for the researchers at Canadian institutions whose dedication and ingenuity are really driving progress in HD research. And this leads to so many advancements that we're starting to see in clinics and so on. I'm very honored to have the opportunity to hear about Dr. Hendricks' work today, and I'm very much looking forward to considering how this could impact families impacted by HD, not just across Canada, but hopefully around the world for many years to come. If during the presentation there's something that you'd like some more clarity on, or if there's something you'd like to learn more about, we really encourage you to submit any questions for Dr. Hendricks via the chat function or the Q&A. I'll be checking them throughout the meeting, and then I'll aim to address as many of them as possible during the Q&A portion after the presentation. So without further ado, let's turn it over to Dr. Hendricks, and thank you all for being with us here today.
Yeah, thanks, Alexander, and the Huntington Society of Canada for the opportunity to tell all of you a bit about the work that we've been doing in the lab. I really appreciate the opportunity, and thank you for organizing. Let me share my screen. All right, so yeah, today I'm going to talk a bit about our work looking at Huntington, both in terms of its roles in regulating and coordinating intracellular transport, and as well as looking at Huntington as a phase separating protein and how Huntington can form these droplets like you see here on my slide, and how those droplets might affect its function and how that might be modulated by polyglutamine expansions. And so, yeah, just to sort of on this title slide, I'm showing a few movies that we took in the lab of different types of intracellular cargoes moving around in neurons. And so you see on the top here, this is a neurotrophic factor called brain-derived neurotrophic factor, and we're watching it move along here at the axon of a neuron in culture. And on the bottom there, we're looking at a different cargo. These are lysosomes. And then, yeah, we'll talk a bit more about these droplets and what they mean a little bit later on. So, yeah, the main focus of our lab is really to understand how do things get moved around in cells, and how are the motor proteins that drive that transport, how are they regulated, how are they controlled in the cell. And so the two main classes of motor proteins that are involved in intracellular transport are these two proteins that I'm showing movies of here. So we have kinesin on the left and dynein on the right. And so kinesins, those are the cargo, or so those are the motor proteins that move things from the cell center outwards towards the cell periphery. And dynein is the motor that moves everything from the cell periphery back towards the nucleus. And as you can see, these motor proteins work by taking the energy that they get from hydrolyzing ATP. ATP acts like the fuel for many active processes in the cell, and converting that to movement along a microtubule track. So the microtubules are polymers that form the tracks that things move along in the cell. And you can see here that the way these motors work, they're sort of like the engines of biology, right? They take in fuel and they convert that to mechanical work or movement, right? And so yeah, we can do things like watch individual molecules of these motors move along. So you see these little dots moving along, or individual molecules of kinesin or dynein. And you can see they very nicely like to walk along those tracks, and that's how things get moved from one place to another in the cell. And so if we think about that in sort of the context of a neuron, right? There, we have many different types of organelles and cargoes that need to get distributed properly and moved around properly in the neuron. We have things like mitochondria, which are organelle that provides the ATP for all these other active processes in the cell, and they need to get properly distributed in the neuron. We have like signaling molecules that need to either move from the axon terminal down to the nucleus or back. And all this occurs along the microtubule cytoskeleton. These microtubule tracks that the cargos move along, and they're moved by kinesins and dyneins. And typically what we have is we have both kinesins and dyneins attached to a cargo. And the reason for that is, I don't know if you noticed in those introductory movies, but many of these cargos are moving bidirectionally. They move in both directions along the microtubule. And kinesins only move outwards and dyneins only move inwards. And so to get a cargo that can move sort of in both directions, we need both classes of those motor proteins. And then the way that the cell can then target one of these cargos outwards or inwards is by turning on or off these different kinesins and dyneins. And so you can imagine if you turn off dynein, then the kinesins would take over and that cargo would move out towards the axon terminal. And if you turned off the kinesins, it would move in the opposite direction, right? Now, these motors move along in 8 nanometer steps, right? But if we think about a neuron, right, a typical, say, motor neuron in a human, the motor neuron that runs from kind of the base of your spine down to your toes, that's about 1 meter long, right? And so given that they take about 100 steps a second, that means like we're thinking about to traverse from the cell nucleus out to the axon terminal, that's on the order of 2.5 days or something like that. So it's a real challenge for intracellular transport. And in fact, we often see that mutations or defects in this machinery that drives transport, either the motor proteins themselves, the microtubule tracks that they move along, or what we'll talk about later, these adapter proteins that hook the motor proteins to the cargo, often results in neurodegeneration, because even though those mutations are happening in all the cells, the neurons are particularly reliant on intracellular transport. And so we see like some examples of this, right? The lab also looks at Alzheimer's disease. Alzheimer's disease is correlated with misregulation of this protein called tau, and tau binds along these microtubules, stabilizes microtubules, and affects transport along them. Also, we have many mutations in these kinesins and dyneins that drive different neurodegenerative diseases. For instance, familial ALS, there's a whole class of familial ALS that's caused by mutations in kinesin. So it's a sort of general theme that we see that defects in the transport machinery, these motor proteins, the tracks, or the adapters, are correlated with neurodegenerative disease. And in fact, that's how we started getting interested in Huntington, because Huntington is one of these scaffolding proteins or adapter proteins that hooks motor proteins to different cargos in the cell. And so, this sketch is a little bit messy, but basically what it's showing is that Huntington interacts with different sets of cargos. So it interacts with things that are involved in degradation in the cells. So these are sort of like the trash machinery in cells, like lysosomes and autophagosomes. They take things that need to be degraded and recycled. And Huntington interacts with those. It also interacts with signaling molecules that are involved in taking think something like BDNF or some other signal that needs to be processed, or also things that are moving out of the cell and being secreted. And in all these cases, what Huntington is doing is Huntington is binding to the cargo and then binding to these adapter proteins that then recruit the motor proteins to the cargo. And what's really interesting about Huntington is that Huntington interacts with lots of cargos. That's relatively rare. Most adapters only interact with a specific set of cargos. And the other thing that's really interesting about this is that it interacts with all the different classes of motors. So it interacts with both kinesins and dyneins, and it also interacts with this other class, which I didn't talk about yet, which are myosins that move along actin filaments for very short range transport. And so in that way, Huntington is poised really at a place where it can regulate both the direction of movement along microtubules by controlling dyneins and kinesins, but also switching between microtubules and the actin cytoskeleton, which tends to sit around the exterior, the cortex of the cell, the outside edge of the cell. Right? So the first question we really wanted to ask kind of coming into this field was, well, what effects do these polyglutamine expansions, so this is the, the mutation that is causing Huntington's disease, what effect do those polyglutamine expansions have on the transport of different cargoes? Because we know from previous work that, again, Huntington interacts with a wide variety of different types of cargoes and also different motor proteins. And luckily, about the time that we were thinking about taking on this project, Mahmoud Pilati, who is an investigator at the University of British Columbia, had just developed, or had developed a few years previously, these embryonic stem cell lines, which he had engineered the huntingtin in those lines to express that huntingtin to have different lengths of polyglutamine repeats. So those lines were completely isogenic, meaning completely genetically identical, except for the huntingtin gene. And the huntingtin then ranged anywhere from 30 repeats, which is associated with normal huntingtin or wild type huntingtin, up to 81 repeats, which would be associated with early onset disease. And so the great things about these stem cells is that we can then take these stem cells, culture them, and then differentiate them into neurons. And so now we have a human neuronal model where we can really isolate the effects of polyglutamine expansions in Huntington, because everything else should be identical, right? And that using these lines, then we could then start to tease apart, well, what are really the effects of Huntington versus other effects that might come about. And so from previous work, a lot of it done in the Fred Sadu's lab, the hypothesis had come out that the defects in Huntington could be due to impaired BDNF transport. So BDNF is known as brain-derived neurotrophic factor. It's a signal that neurons send to each other to say, yes, we're in a happy environment and we should continue to function and stay alive, right? And so lack of BDNF signaling is associated with neuronal death, right? And what's really kind of interesting in Huntington's disease is that the striatal neurons that are primarily affected by Huntington's disease don't actually manufacture their own BDNF. So they are reliant on the cortical neurons to make the BDNF, for the cortical neurons to then transport it down their axons, secrete it out, and then be taken up by the striatal neurons. And so the hypothesis there was, well, if there's a defect then in the transport of these BDNF vesicles in the cortical neurons, that would lead to impaired BDNF signaling in the striatal neurons. But at the same time, we were interested in both BDNF transfer, but also we knew that Huntington was also interacting with all these other cargoes. And so we started off with really just sort of looking at different cargoes in the neurons to see, well, what is the effect of Huntington? And we started with looking at two cargoes. We looked at this BDNF that I just mentioned, and then we also looked at lysosomes, which are part of, again, the degradative machinery. They're part of taking the things in the cell that need to be degraded and recycled, and putting them in an acidic environment, well, then they'll be broken down and then recycled, right? And so we started by looking at these lysosomes and how they moved in these neurons. And so here we're looking at the 30q wild type neurons. You see just a snapshot of, here are the small little lysosomes in that axon. And then what we're doing is we're depicting the movement on what's called a kymograph. And so what's happening here is that we've drawn a line through this axon and then projected the intensities over time. And so what you see here is that if we have a line that's moving off to the left in time, that is a cargo that's moving back towards the cell body. So this is a what's called retrograde or movement back towards the cell body. If we have a cargo that's moving out towards the neurite tip, which would be off to the right, then you'll see that those are marked kind of in pink here. So a slant is going this direction. And then if a cargo, a lysosome, is just staying in one place over time, then that just looks like a vertical line, right? Because over time, the position isn't changing. So we just see this vertical line, right? So you see, yeah, here are a bunch of retrograde trajectories. and some anterograde trajectories. And what you see for lysosomes in the normal case is that typically there's sort of an even balance of retrograde and anterograde motion, so motion in both directions. And what was kind of interesting to us is that as we looked at neurons expressing expanded Huntington, what you can see is now there's a shift. Actually, you see kind of there's more pink lines as we shift to the right and fewer green lines as we shift to the right, right? And so we quantified that to try to understand exactly what was happening. And so there, if we look at the number of cargos, that's not really changing very much for the different lines or the different polyglutamine repeat levels. But if we look at the ratio of outward movement to inward movement or anterograde to retrograde movement, what we see is that as we increase the polyglutamine expansion, that there's more and more anterograde movement, right? There's more anterograde movement compared to the 30Q line. And so that indicates to us that there's some misregulation of that transport where cargos actually aren't going to the place they're supposed to go. They're supposed to primarily sort of go both directions and move towards the cell center over time. And here we see they're actually moving out towards the neuronal tip, right? And so then we also looked then at these brain-derived neurotrophic factor vesicles, and there again we're looking again at these small cargos as they're moving along the axon, and we've drawn out maps of that movement the same way we had done before. You see these cargos are very bidirectional, right? They're moving really back and forth a lot, which is driven again by the kind of interaction between the kinesins pulling one way and the dyneins pulling the other way. And there, if we quantify that transport, one thing that's kind of interesting that we see is that at moderate expansions of the polyglutamine, we see actually an increase in the number of BDNF cargos, whereas at the 81Q, there's a small decrease. And then if we look at the direction of movement, there what we see is if we look at the distance these cargos move, as we increase the polyglutamine repeat length, we get longer and longer runs, or it moves a longer and longer distance towards the nerve terminal or out towards the nerve terminal in the anterograde direction. But in contrast, the retrograde or back towards the cell center movements are getting shorter and shorter. So here again, we do see this clear kind of misdirection of BDNF transport. We're getting more and more movement out towards the axon terminal and less movement towards the cell center. And so that would be expected to have defects then in the signaling, right? Because that's going to affect the number then of those cargos getting out to the nerve terminal. So we were very curious to understand, well, how is this happening? What's causing this shift in transport to more transport out towards the nerve terminal? And so what we did then is we took cells where we had these BDNF cargos, we broke those cells open and isolated out the cargos, and then looked at each individual BDNF vesicle and counted the number of motor proteins on there, the number of huntingtins on there, to sort of see what's different between these different sets of cells, right? And so the first thing we wanted to make sure of was that the endosomes that we looked at, these cargos that we looked at from the different cells, had the same identity. So we looked at a bunch of markers that mark different groups of endosomes. So this would look at early endosomes, late endosomes, and lysosomes to sort of see what fraction were in these different groups. And there we see, well, the fraction that are in those groups is quite similar between them. But then when we started to look at, well, actually what members of the transport machinery are on these cargos, that's where we actually started to see a difference. And so what you can see here is if we compare the 30Q neurons, or the endosomes isolated from 30Q neurons versus 81Q neurons, first, the fraction of those vesicles that have Huntington on them is much lower. So it goes from about 80% to 35%. Also, there's fewer of those that have this Huntington-associated adapter protein called HAP1, which interacts with Huntington, but also kinesin and dynein. And yet we had more of them that had kinesin on them. And then we can also use this tool of quantitative fluorescence to be able to actually count how many of each of those proteins, how many each of those motors are on the cargo. And what we see there is also quite interesting, where we see of the cargoes that have huntingtin on them or have this protein on them, we have about equal numbers of huntingtin, but there's about 4 times as much HAP1 on each of those cargoes. And that in turn recruits about 2.5 times more dyne or more kinesin. And now this is kind of starting to make sense, because remember, the kinesins are the motor proteins that pull out towards the nerve terminal. And if we're getting more of those kinesin motor proteins on the BDNF cargoes, that would then explain why we get this shift towards more outward motion, because there's actually just more motor proteins there. So you can imagine that team of kinesins is getting bigger, and they're kind of winning that tug of war to drive more cargoes out towards the nerve terminal. And so I think that now gives us kind of a molecular mechanism for where these defects in transport occur. So now we know actually, well, we know that the polyglutamate expanded huntingtin is affecting the adapter proteins and then the motor proteins as well. And now we can start to figure out, well, what disruptions are really causing the changes in transport that we observe. And so the next part of this that we're really interested in is that, again, this is kind of a common theme in neurodegenerative disease, but often the mutations are present from birth, but the symptoms don't manifest until much later in life. And so, what is really the trigger for triggering the onset of disease symptoms? And one of the things that we've been really interested in, broadly in neurodegeneration, but in particular in Huntington's as well, is this seeming connection between inflammatory responses, so responses to some sort of infection, and neurodegeneration. And so we started thinking about this in terms of Huntington, and actually there had been some prior work showing that if we look at different markers for inflammation, so these are different cytokines that we know are upregulated when there's an inflammatory response. What's really interesting is if we look even in Huntington patients that is prior to the experience of symptoms, what we see is actually there is an enhanced inflammatory response that's present there. Many of these markers are enhanced over the control. And if we look as we look farther and farther into disease progression, those markers tend to increase. And so we wanted to see, well, what is the effect on transport of both the polyglutamate expansions, but also this neuroinflammatory response? And so the way that we modeled that is we treated the neurons with this neuroinflammatory cytokine called interferon gamma, which then elicits an inflammatory response in the neurons, and then looked at transport again. And so, and then now compared the 30Q case, so the normal number of polyglutamate expansions with a disease mutation, right? And what we see there is quite interesting, because what we see is that if we compare the 30Q neurons and then in the neuroinflammatory condition, what we see is actually the number of lysosomes increases. If we look at the 81Q neurons in normal conditions, they're actually already at that elevated level of lysosome. So it almost looks like here that the polyglutamate expansions are actually acting like a stress or like a neuroinflammatory response, and that we increase the number of lysosomes. And then if we subject now those 81Q neurons to neuroinflammation, we get an even higher number of lysosomes. So we could kind of think about this as the neurons are getting sort of two insults, right? They're getting the sort of stress from the polyglutamate expansions, and then they're getting increased stress from this neuroinflammation, right? And we can see there that actually affects the direction of transport as well, where we get this sort of shift towards more plus end directed transport. And that shift is even enhanced in the 81Qs, although if we compare the 81Q neurons with the neurons under stress, that's quite similar between the two, right? So then we were interested, okay, let's look at the BDNF transport. So we did the same thing there with BDNF. And what you can kind of see already kind of from the raw data is that There is definitely an effect of the polyglutamine expansions on the number of BDNF cargoes. So you see it's much lower here in the case of the stressed polyglutamine expanded neurons. And actually, if we quantify that, we see exactly that. So for 30Q neurons, stress induces a slightly higher number of BDNF cargoes. For 81Q neurons, it's actually decreased, right? But then it's even sort of more interesting when we look at the direction of transport. So here again, we're looking at these run lengths, how far do they move? And you can see that for the 30Q neurons, if we stress those neurons, we get longer runs in the anterior direction, shorter runs in the retrograde direction. Now, if we do the same thing for the 81Q neurons, we actually get shorter runs, much shorter runs in the anterograde motion, and no change in the retrograde or back towards the cell body. And so what's really interesting here is that it looks as though the 30Q neurons and the 81Q neurons are actually having opposite effects of neuroinflammatory stress, right? So here we wanted again to sort of get some more insight into, well, what's actually changing on the cargoes themselves? What is driving these changes? And so we use this technique called DNA paint. And I'm going to take just a second to try to explain sort of how this DNA paint works. And so typically, when we are trying to image a specific protein in the cell, what we do is we use antibodies that will recognize that protein of interest directly. So that here's this antibody, here's the protein of interest, here's our antibody. And typically we would then stick a fluorophore directly onto the antibody so that we would be able to visualize it. And that's what actually lights up under the microscope is this fluorophore, this little depicted as the little pink dots here, right? In DNA paint, rather than hooking the fluorophore directly onto the antibody, we instead use this small strand of single-stranded DNA, and then we hook the fluorophore to a complementary strand of DNA. And so the reason this is useful is because those two complementary strands of DNA will now bind transiently. So they'll bind and unbind and bind and unbind and bind and unbind. And the reason that that's useful is because we can now then correlate the frequency at which we see imager strands binding to the number of proteins that are there, the number of these docking strands that are there. Because if you have imager strands binding to one docking strand, you get one frequency. And then if there's two, now you get twice that frequency. And so you can directly correlate then the frequency of the blinks to the number of proteins that were there. And so you can, here's an example of what these movies look like. So you can see on the left, we've labeled the BDNF cargoes. So you see these bright dots are BDNF cargoes on the left. And then if we look at HAP1, so this is that Huntington associated protein 1. If we look at that using this DNA paint, so here again, we see these are 3 different axons that we're looking in. And you see there are many Huntington or HAP1 associated vesicles or cargoes along the axon. And each one of these is blinking because those imager strands are attaching and then detaching, attaching and detaching. And for each of those, then we can zoom in and look at the frequency of this blinking to count how many HAP1 molecules were actually on that specific cargo. And so if we use that technique and we look first at the effect of neuroinflammatory stress just on the 30Q neurons, what we see there is that stress tends to increase the fraction of those vesicles that are positive or that have kinesin and dynein on them. It doesn't really change the number of Huntington or HAP1 molecules there, but it does increase slightly the number of motor proteins that are associated with those cargoes. If we count then the number of those motor proteins, that seems to be quite similar, right? So there's actually relatively minor changes, and if anything, there's slightly more motors that are associated with those cargoes, okay, to drive their transport. Now, if we look at the 81Q neurons, it's exactly the opposite, right? If we look, there are fewer dyneins on those cargoes, there's fewer kinesin, there's fewer HAP1, and there's fewer cargoes that have HAP1 and Huntington on them as well. So there are fewer of the cargoes actually have Huntington or its related transport machinery there at all. And then if we look at the number of molecules that are there, the number of hap1s decreases and the number of huntingtins decreases by about 3 times, or a factor of 3, right, for the huntingtin, and about a factor of 2 for the hap1. And so this is kind of indicating that in the context of neuroinflammatory stress, the polyglutamine expanded huntingtin is just unable to actually be recruited to the cargos, and in turn unable to bring the motor proteins there. And so it's just sort of just summarized here, where we can see, okay, the effect of neuroinflammatory stress on control neurons is that primarily we get these green boxes, meaning that there's an enhanced recruitment, right? Whereas if we look at 81Q compared to control, in some cases there's enhanced, in some cases there's decreased enhancement. But if we look at 81Q neurons in the presence of neuroinflammatory stress, now there's just fewer both of Huntington-associated proteins, but also the motor proteins that are driving transport. And so again, this sort of looks like there's sort of now multiple insults that the polyQ expanded neurons are having to deal with. First, they have the polyglutamine expansion, which is causing defects, and then on top of that, if they're trying to respond to cellular stresses or neuroinflammatory stresses, they're unable to really recruit the proteins to the cargos that need to be there for them to be transported properly. And so I'm just sort of summarizing that here, where we think that possibly what's happening is that typically we would have an early endosome, and it would later sort of mature into a late endosome, where this hap1 would help it transport and be allowing to progress properly through the cycle. Whereas under stress, we get moved to a different pathway, right? Which either might have different Huntington-associated proteins, or, as we're seeing in some cases, Huntingtin is just not able to be recruited to the cargo at all, right? All right, so that's sort of was a summary of kind of where we're at and sort of thinking about Huntington's role in intracellular transport. And so I'm going to sort of shift gears a bit at this point, because Huntington has very well described roles. Huntington protein has very well described roles in, again, moving things around, as we've talked about in intracellular transport. But the other side of Huntington is that we also know that it has strong effects on transcription and RNA processing. And actually, if we look at what pathways are really downregulated in Huntington disease patients versus controls, what we see is there's a lot of things involving RNA processing, and DNA repair, right? So there's clearly a role here in transcription and RNA processing as well, right? And that role, I think, has been a bit harder to understand in terms of what proteins are really driving that regulation. How is that working? But some clues started to come out recently from a collaborator. This is from Rachel Harding's lab at University of Toronto. And Rachel's lab noticed that when they purified Huntington, or when they pulled out Huntington, what they often found was that there were many RNAs that were specifically interacting with Huntington, including RNAs that are known to have a role in transcriptional control, right? And then if they looked in cells to see, well, what does Huntington localization look like? And here specifically, we're looking in the nucleus of the cell, so the part of the cell where the DNA and everything is contained, right? What we see is actually Huntington is present in the nucleus of the cell. It's forming these kind of globs, I'll call them for now, right? Maybe we'll call them condensates in a second. And some of those are actually, we can see that if we look for specific RNAs which are known to interact with Huntington, we see that in some cases those correspond to these Huntington blobs in the nucleus as well. And so it's clear that Huntington is in the nucleus. It's forming these kind of blob-like structures, and it's there. It's also interacting with RNA. And so this was kind of the question that we started with, was, well, what's Huntington doing there? How is this working? And in many other contexts, in recent years in cell biology, we've seen that proteins often form these blob-like structures or droplets in order to mediate their function. And it's a way for the proteins to concentrate and bring other things together, and to promote different biochemical reactions, etc. Many times these proteins are small and largely disordered. And so it was a little bit of a puzzle because we had, again, from Rachel Harding's group, we had structural information about what huntingtin looks like. And huntingtin is largely structured. It's a very large protein, 350 kilodaltons, and largely it's structured in this very tight structure, right, with these heat domains, and in complex with its obligate binding partner, HAP40. But if we look closely at huntingtin, there is this intrinsically disordered domain, which means a very floppy, very flexible domain. And those intrinsically disordered domains have been implicated in driving this formation of droplets, or what we call condensation. And so we thought, okay, well, let's look to see, does huntingtin in fact form these droplets? And so with Rachel's group, Rachel Harding's group sent us purified full-length huntingtin along with NEAT1 RNA, this RNA that's been shown to interact with. And when we put that under our microscope, what we see is very clearly, we see these droplets are forming, right? So these droplets form either in the absence of RNA, but if we add RNA there, you can see that that affects the size and distribution of those droplets. So it's clear that the RNA is actually interacting with the huntingtin in these droplets as well. And what was quite interesting to us as well is that if we compare the different polyglutamine lengths, so here we're comparing 23 and 74, what we see is that there's a clear difference in the shape and the size distribution of these droplets. And so it's clear that there's a direct effect of the polyglutamine expansions on this condensation as well. And so we started then to delve into this a little bit. And Rachel's group created a version of huntingtin that was missing that intrinsically disordered domain near the N-terminus. And what was quite interesting, we look at that, it does not form droplets. So we don't see droplets forming there. So it's clear that that section of the huntingtin is really important now for driving that phase separation. We also use some tools then to be able to quantify what are the mechanics and dynamics of these condensates or droplets. And so one of the techniques we can use for that is called fluorescent recovery after photobleaching. And so in this case, what we're doing is that the huntingtin molecules have been labeled with a fluorescent protein. And so that's what we're visualizing here. And then what you're going to see is that when I play the movie, we shine a very bright laser on one part of the protein and convert all those molecules to a dark state. So they're no longer fluorescent, they're no longer giving signal. And then what we can watch is we can watch then as those dark molecules diffuse out of that region and bright molecules diffuse in. And so here you see two regions where we photobleach, and then you can watch then as the proteins are mixing within and between the condensates, right? And so that movie was for 23q huntingtin. If we do that same technique now for expanded huntingtin with 54 polyglutamine expansions or polyglutamines or 73, you can see, I think, already that the dynamics of how quickly those molecules are rearranging within that liquid-like condensate are much different, right? And in fact, we can quantify that. So if we compare the intensity in that region that we've bleached or made dark to outside and look at the dynamics of that, what we see actually is that that follows what's called a compressed exponential fit. And so in a simple phase separation system, that would be just a single exponential. It would look just like a single exponential. But here we see that that is sort of, well, it was compressed. And this term that we're seeing here, this beta term, that gives us an idea of how interconnected the different molecules are in that condensate. So as that goes up, that means the molecules are more and more interconnected. As the time scale goes up, that means that things are moving slower, right? And so we can see here, we can compare this now, whoops, for, again, 23, 54, and 73q. And what we see is that the time scales for that rearrangement go up quite clearly as we increase the polyglutamine expansions. And also this beta term, or this term indicating the connectivity of the proteins within that network, also increases. So we want to look at this a little bit more closely. And so we use this technique to look at how two of these droplets will fuse together. And so you can imagine that if these two droplets are behaving just like liquids, so like oil and water, if two little droplets of oil were to come together, then what we would expect is that eventually they would fuse. The surface tension of the droplet is driving their fusion, and they would fuse into another spherical droplet, right? And so in this case, the way we're doing this is that we have a focused laser beam called an optical trap, which we can hold the two condensates in, hold those two droplets in, and bring them close together, then watch as they fuse. And so, yeah, that's exactly what we see for the 23q Huntington, is that we see those two spherical droplets come together and form another big spherical droplet, right? Now, if we do the same experiment for polyglutamine expanded Huntington, what we see is that the surface tension starts to drive their fusion. But then what happens is that the springiness of those condensates starts to fight their fusion. And then you get this sort of balance of forces of the surface tension trying to drive them together, and the elasticity resisting that motion. And so then you get a rest, right, where you get not a spherical condensate. Right now you get some sort of deformed condensate. And so we can actually use those shapes to then back calculate out what are the mechanical properties of those condensates, and looking at what is the effect of elasticity versus viscosity, right? Or liquid-like versus solid-like. And so we can quantify that. So if we quantify that for, here again, the 23 versus the 73q Huntington. So here, if we were to follow the x-axis here, that would mean it's a purely viscous system. And as we move up, that means we're getting more and more elasticity, or more springiness, more springiness basically. And so you can see that there's a clear difference in the properties of the 23 versus 73q, where the 73q is much more elastic, much springier, right? So we see very clear differences in the 23 versus polyQ expanded Huntington. But one of the questions is, well, what is the function of these droplets? And so there we use this technique called Raman. And so what Raman does is it gives you sort of a signature of that protein. And so from there we can quantify the amount of water that is in each of these condensates or droplets, the amount of protein that's in there, and then what is that we get some clues from this fingerprint region about what is the structure and of the protein, and how are those interactions between proteins changing. And so what we wanted to use this tool for was to see, one of the hypotheses that you can make is that the formation of these droplets of Huntington is potentially on the pathway to forming solid-like aggregates of Huntington, right? So we get first liquid-like droplets where we have Huntington concentrated in these droplets, and then that might actually nucleate or spur or trigger the formation of solid-like aggregates, which we know could be pathogenic, right? And so what we did is we monitored then the Raman spectra for both the normal and the 73q Huntington over 7 days. And what was really interesting there is that basically not much happened. So you can see, if we look at the region which tells us about the sort of structural interactions of the Huntington, that stays almost exactly the same over time. Whereas maybe, over time we're losing a little bit of water out of the condensates. So a little bit of the water is coming out of the condensates as they age, but the structure of Huntington is staying quite similar. And so that argues that actually the Huntington that's in these condensates maintains a very stable structure over time. And these condensates might actually be a way to help Huntington maintain its structure over time rather than become misfolded and forming aggregates. So now we're sort of, starting to think more and more about, we see that there are these very clear changes in the mechanics and dynamics of these condensates that correspond directly to the polyglutamine expansions. And that's really exciting to us because, it's really kind of been a puzzle. Often when we look, say, at the structure of uh, normal huntingtin versus mutant huntingtin, it looks very similar. If we look at the protein-protein interactions that are described for normal versus mutant huntingtin, those look very similar. So what is the root cause of the problem with the polyglutamine expansions? And here we have now an observation where we can directly see a change in the mechanics and dynamics as a function of the polyglutamine expansions. And so I think there's a couple different ways that this could potentially come into Huntington function. One is that clearly these condensates in the nucleus are likely interacting with RNA and likely other transcription machinery in order to regulate its activity, right? If you're in the condensate, now you're bringing all those things together, and that could enhance its activity, or it could sort of be sequestering it. Also, we could think about these condensates in the cytoplasm serving as sort of a pool of properly folded huntingtin that then the huntingtin can easily come in and out of these condensates to interact with cargos or to interact with RNA now in the cytoplasm. So you can imagine now, if you start to make those condensates more stiff, more solid-like, and now you're decreasing the amount of exchange that can happen from the condensates and out, and also you're making these condensates more stiff, that would then have a downstream effect on all the transcriptional regulation that's happening within this condensate, as well as the ability of huntingtin to now interact with other structures and other things like vesicular cargos in the cell that it needs to interact with. So yeah, that's the main gist of what I want to talk about today. But I want to definitely thank all the people and acknowledge all the people that really did the work. And so the studies on transport in Huntington that we talked about earlier were really driven by two graduate students in the lab, Emily and Brooke, and along with a lot of help from a postdoc, Muriel, and help from Daniel as well. Muriel driving the sort of stem cell work in the lab, and Daniel the single molecule biophysics. The work on neuroinflammation was all led by Lale Gersu, and the cells again were developed by Mahmoud Palati. Mahmoud Palati has been a really great collaborator in teaching us how to use those cells and helping us to characterize them. Ben Guerin and has really driven the condensate work in the lab with some help from Lilian, another master's student in the lab, and that has really been only able to do through a tight collaboration with Rachel Harding's group and specifically Eileen Haidari and Rachel Harding's group that's able to produce this full-length protein for us to work with, has all this biochemical characterization that really informs a lot of that work. And lastly, Matt Harrington here in chemistry, I think has helped us with all those Raman measurements. So with that, I'll very much appreciate your attention and again the opportunity to speak, and I'd be really happy to answer any questions.